this lecture focuses on meiosis and sexual reproduction. And the basic idea is this, that we're going to talk about this cellular process that cuts the chromosome number in half. There is no other use for meiosis besides sexual reproduction. And so you're familiar with the fact, with diploid and haploid cells, you know that parents are diploid. Meiosis cuts that chromosome number in half, so you go from 2N to N, diploid to haploid, and then fusion of those haploid gametes results in a zygote that once again is diploid. Again, a zygote is a single-celled embryo, Now, some of this is going to be review for you, I know, but we'll talk briefly about the idea of homologous pairs of chromosomes. Now, we understand that in every cell of our body we have our complete set of DNA, our complete genome, and so we have these homologous pairs of chromosomes that are located in each cell of the body. Every cell in our body, in other words, is diploid, except for the gametes, which are haploid. Humans have 23 different types of chromosomes, as you're well aware, and if you look at each of those pairs of chromosomes, as we have on the screen, as I'm pointing with a little cursor, what I thought would be the cursor, because it turns into a weird little shape when I put it over the picture, then we see like pair number 14, pair number 15, pair number 16, pair 20, and those are homologous pairs of chromosomes. One of those came from your father, one of those came from your mother. The homologous chromosomes, as you well know, carry alleles. Alleles are alternate forms of a gene. And so if we look at, for instance, as I point here, the left homologue for pair number 14, we might have red hair on that chromosome. And if we look at the other one, we also have a gene for hair, but it's not necessarily red hair. It could be brown hair, blonde hair, or so forth on the homologue. Now that's a bit of an oversimplification because, as you know, hair color is coded for by multiple genes, but it seems to be an example that kind of gets the point across, I hope. Now, homologous chromosomes are the same length, and that will allow you to match them up. You can tell which chromosomes go together. And is there anything else we need to say about them? I think that pretty well covers homologous chromosomes. They're not all the same size. They contain alleles. You understand that there are 20,500 genes approximately that make up the human genome. So each of these chromosomes would have a large number, close to 1,000, maybe over 1,000 genes located along its length. Now, just like in mitosis, we see that we have these stages that occur within the process of meiosis. In fact, in meiosis, we divide it up into two different sets of divisions, meiosis 1 and meiosis 2. And we're going to talk about some special events that occur in each of these. But the basic idea is this, and it's illustrated down here in this picture in the bottom portion of our screen. So in meiosis 1, this will be extremely important to you if you can remember this and if you understand this. So in meiosis 1, the homologous pairs of chromosomes line up. See where I'm pointing there on the screen? You see the homologous pairs lining up. You see a red chromosome here and a blue one there. Red perhaps indicates it came from the mother. Blue indicates it came from the father. Obviously, they're not really red and blue, but the book has color-coded them for your benefit. You can tell they are two parts of a homologous pair because of the length. These two chromosomes are exactly the same length, and these two chromosomes are the same length. So in this example, we have two pairs of homologous chromosomes lined up together, and those chromosomes are going to split in meiosis 1, as is depicted here where I'm pointing, to become two separate cells, and therefore we go from diploid to haploid. So meiosis 1 takes us from diploid, where I'm pointing here, two copies of each homologous pair of chromosomes, to haploid, where I'm pointing here. Now there is a red a red chromosome and a blue chromosome here, but you see there's only one of the short pairs here. See one half of the short pair, one homologue there, and then there's one of the longer homologue, one homologue of the longer homologous pair. So there's a longer blue chromosome there and a longer red chromosome there. And so this cell is on it's a pathway to become an egg or sperm, as is this cell. Now in meiosis 2, as you can see, what happens is the duplicated chromosomes split. So if I go back over here and point, we have two sister chromatids composing this particular chromosome. And now follow the pointer to where it's going at this particular point in time. So that homologous pair I just pointed to is split. One sister chromatid, which is in and of itself a complete chromosome, is there. The other half of that chromosome, which of course is also a complete chromosome, is right there. So this cell I'm pointing to now has become this cell and this cell. This cell that we see here became this cell and that cell. And so the basic idea of meiosis, if we can summarize, is that in meiosis 1, the homologous pairs of chromosomes split. In meiosis 2, the sister chromatids themselves split. You actually probably know this very thoroughly from your lab this Thursday, but just to reemphasize, that's exactly what happens. Let's go on to our next slide. So we see an overview of what's occurring in meiosis in 1 here. And now one interesting thing we want to note is that something referred to as synapsis that was spelled out on the last slide occurs in prophase 1. Synapsis is illustrated right here. It's when the two chromosomes become closely associated with one another and connected via a protein lattice. It's at this point that something called crossing over occurs, which we will explain in a moment or two, a couple slides later. So that occurs in prophase 1 of meiosis. In metaphase 1 of meiosis, this is a question you'll commonly be asked over the next couple weeks and up to the final and then on in your biology career. So what lines up at the metaphase plate at metaphase 1? And you will always say homologous pairs of chromosomes line up. See where I'm pointing right now? This is the metaphase plate in metaphase 1. And you'll see one homolog on one side and one homolog on the other. So we're using the term homologous pair to refer to both of these chromosomes, and we're using the term homolog to refer to one or the other chromosome. So this is one homologous pair, this is the other homologous pair. They're going to separate in anaphase 1 of meiosis. In telophase 1 of meiosis, the nucleus is reforming, cytokinesis is occurring, and then at the end of meiosis 1, we have two separate cells. And if I was in class with you, I'd ask you, are these cells diploid or haploid? And you would answer, they are haploid. And I know they're haploid because as I look at these chromosomes, I see I have a short chromosome and I have a long chromosome. And that means that this homologous pair, the short homologous pair, split. One's there, one is there, and then the long homologous pair split. One is there, and one is there. Very good. So we go to the next slide, and we see what happens in meiosis 2. So in meiosis 2, we notice that we go through prophase, which is similar to the previous prophases we've discussed, but then look at metaphase. So in metaphase of meiosis 2, I will ask you frequently, what lines up at metaphase of meiosis 2? And you will always answer, the chromosomes themselves line up. Or you might say the sister chromatids line up. So you'll notice that just like in mitosis, the chromosomes themselves are lined up along the metaphase plate to which I'm pointing at this particular time. Then in anaphase, what splits in anaphase 2 of meiosis? And you will always say the sister chromatids split. The sister chromatids are pulled apart in anaphase 2 of meiosis. We notice that in telophase 2, the nuclear envelope is reforming, cytokinesis occurs, and we end up with 1, 2, 3, 4 daughter cells at the end of this process. So to summarize then, we have these two types of divisions that we've talked about in the past week in both lecture and lab. And so we have meiosis, which requires two nuclear divisions, involves crossing over, and we notice that we have 4 daughter cells that are produced in meiosis, and it's only used for sexual reproduction. The centromeres do survive anaphase 1, as you notice in meiosis. So the centromeres, as you recall, are holding the two sister chromatids together. You remember it's not until meiosis 2 that the sister chromatids are pulled apart. And of course, meiosis cuts that chromosome number in half. Mitosis, we won't go back over all those, but you recall from your last lab and the last chapter that mitosis preserves the chromosome number and produces cells that are identical to parent cells. In meiosis, because of crossing over in random assortment, our next topics, then we see that we do not have cells that are identical to the parent cells, aside from the fact that the chromosome number is cut in half. So there are many reasons in meiosis that that the cells are not identical to parent cells. All right, so we won't spend much time on this, but this is a nice review. It really is a good review that I would encourage you to look at that's found in your text. So look at this. We have meiosis 1 on the top, and so we ask what lines up. The part I like the most is what lines up at metaphase 1 of meiosis. And you will always say the homologous chromosomes. There where I'm pointing is the metaphase plate. The homologous chromosomes are located on either side. And look, here is mitosis, excitingly. And so we notice that the chromosomes themselves are lined up along the metaphase plate in mitosis. Now that does look like meiosis 2, doesn't it? But of course we know that in mitosis the cell is diploid, so all the chromosomes, all 46 of them, are lining up along the metaphase plate. In meiosis, in humans, I'm referring to humans here of course, then we would only have 23 cells lining up along the metaphase plate in meiosis 2, metaphase of meiosis 2. And here we see everything wrapping up here. There's mitosis concluding, and then we see meiosis 2. They actually don't show much of the detail of meiosis 2. But we see that in the end, we end up with 1, 2, 3, 4 cells in meiosis. And are those cells diploid or haploid? Of course, you answer they are haploid. In mitosis, we only have 2 cells, and those 2 cells, as you well know, are diploid. Now, one interesting feature of all this is the following, that in mammals, what occurs when we have gametogenesis, which is the process of meiosis producing the eggs or sperm, is this: that in sperm, shown on the left-hand side, when we start this process, it's just like you would have expected based on what we just said. You end up with 4 sperm that are all viable, that could all fertilize an egg. However, look over at the right-hand side. In the production of eggs, referred to as oogenesis, what we end up with is this. We go through a division that you can see shown on the screen here, and all of the resources of the cell go in one direction. So this is the nucleus that we see here, and we've interestingly thrown away half of the DNA. So you recall that when we undergo this division, we cut the chromosome number in half, and we have to get rid of it when we're producing mammal cells. So we produce this thing referred to as a polar body that's haploid, and the polar body contains the extra DNA that's not going to be used. All the viable DNA is in the nucleus that we see here. Interestingly, the egg doesn't complete meiosis 2 until fertilization, and so in meiosis 2, a second polar body is formed. You realize that those chromosomes, which are duplicated, split. If you have any doubts about that, glance back a slide. So that's the division that occurs in meiosis 2. Now, there would only be 2 polar bodies formed because the first polar body doesn't divide again, right? We have one division, and we produce one viable. We have the first division, and we produce one viable cell, and then we just have the polar body, which is not going to divide. But then, of course, this cell undergoes another division, producing a second polar body, and then only the DNA that remains in this functional egg is going to unite with the sperm to make the actual embryo. So it's an interesting feature of meiosis in mammals, at least. In sperm, we end up with 4 viable cells, but in eggs, the production of eggs, oogenesis, we only end up with 1 egg that's actually going to be fertilized. So we go through 4 divisions, but we have 1 viable cell. Now, most excitingly in all this discussion is the discussion we get to now of crossing over and independent assortment. Now, briefly, let's preface this by saying, let's make an observation in the natural world. So, 2 observations. One observation is that organisms could reproduce in one of 2 ways, and those ways are sexual reproduction and asexual reproduction. And so the first observation is that asexual reproduction is easier to accomplish. Observation number 1. Second observation, asexual reproduction is much less common in the natural world than is sexual reproduction. And so you have to ask the question, why? If asexual reproduction can be accomplished so much more easily, why would organisms reproduce sexually? And the answer is clearly the advantage that's conveyed via genetic variation. So much in biology, as we approach this natural selection and evolution discussion, coming very shortly in future chapters, relies on this idea of genetic variation. And we'll make that very clear as we move forward. But now, what we get to focus on for the next 2 minutes is, or so, maybe a little longer, is the source of this genetic variation in living organisms, which is an exciting idea. The genetic variation comes in two ways primarily: crossing over and independent assortment. First, crossing over. So crossing over occurs when during synapsis, and that takes place, as you recall, during prophase of meiosis 1. So take a look. I have some videos and so forth that we can take a look at in class, and I usually use, but let's use the pictures from our text for now. So if we look at B, then we see a picture of one homolog and a picture of the other homolog, the red one and the blue one, and they are zippered together during this process of synapsis. And so what occurs is that, fascinatingly, the proteins break. And in this case, if you can see where I'm pointing, I hope that you can with that little thing on the screen, cursor, I guess, you notice that the blue one breaks and the red one breaks, but they break at the same point and they trade segments, which is an amazing idea. You're swapping segments. So if you want to personalize this, which might be disturbing for you, think about your parents. So the chromosomes of your parents were scrambled in this manner before they made you. In your father or mother, there was a chromosome from your paternal, let's say your mother, your maternal grandfather and your maternal grandmother, and they are, that's the only place that your mother could have gotten those chromosomes. And so you only got one of those, obviously. You did not get both. But before that chromosome came to you, they were mixed up in the manner that we see here, described as crossing over. And so the chromosome you inherited was not completely from your grandmother, not completely from your grandfather, but it was a combination of genes from both of those people, which I think is an interesting way to look at it. And it provides a tremendous amount of variation, a tremendous amount of scrambling of the genes before the genes are passed on to offspring. So that's the idea of crossing over. What we see in D, as you'll notice, is the chromosomes that are created as a result of the crossing over event. There would, of course, be 4 chromosomes that are produced as a result of the 4 cells. And in oogenesis, 3 of those would be thrown away, only 1 would survive. But we do have 4 chromosomes that are produced. 2 are actually did not undergo crossing over, as you notice here, and 2 of them were the subject of multiple crossing over events. So crossing over, as we understand it, crossing over typically occurs with each pair of chromosomes every time meiosis takes place. It's not an isolated event. It doesn't have to occur, but it almost always occurs, and it is a random event. The point at which the break occurs is random. It's interesting. I'll throw this in. I talk about this in more detail in lecture, but one of the only 3 Nobel Prize winners from the state of Kentucky was Thomas Hunt Morgan. And Thomas Hunt Morgan was a famous geneticist. He received the Nobel Prize for discovering the sex-linked traits, like colorblindness, for instance. He didn't come across that one, but we'll talk about him more on Monday in Chapter 10. But he did a lot of work with this, and he was able, fascinatingly, to map the location of genes on chromosomes based on the frequency of crossing over. There's a bonus question that I will include on the upcoming exam related to that. So how did he map those genes using the frequency of crossing over events? It's a pretty interesting thing. When I was in grad school at UK, we did fruit fly crosses, which is what he used, and we had these huge crosses, and we observed the exact same frequency that he observed. So he was able to, he's operating back before 1900, and he was able to identify the location of genes on chromosomes without any type of sequencing that we have today. He blindly mapped them using the frequency of crossing over, which is pretty fascinating. So overall, then, that's a nice bit of bonus information, but the most important thing that we want to take away is that crossing over is a very effective method of scrambling the genes. Now, the number 2 method that brings genetic variation into offspring is this idea of independent assortment, which is easy to explain and easy to understand. So if we look at the screen, we see independent assortment. And let's go to the left-hand side, left-hand bottom of the screen. So what we notice is this. This is a hypothetical cell that has a diploid number of chromosomes of 6, a haploid number of 3. There's only 3 pairs of chromosomes. So it's possible that in the process of meiosis 1, it's possible that all the chromosomes from the father, which I'm going to say are shown in blue here, line up on the left, and all the ones from the mother line up on the right. I hope we're all on the same page there. Hopefully that makes sense to you. These are homologous pairs that I'm pointing to. Here's one, here's another, homologous pairs of chromosomes. And it's possible that all the chromosomes from one parent line up on one side and the other parent line up on the other. But that's not how it happens. So when the chromosomes line up, the first pair that I'm pointing to here lines up, and we could flip a coin. There's a 50% chance the blue one ends up on the left side and a 50% chance the red one ends up on the right side. But they could just as easily be flipped. There's an equal probability of both occurring. However, excitingly, when the next pair lines up, the arrangement of the first pair has no influence whatsoever on the second pair. So once again, 50% chance that the blue one ends up here and 50% chance that the red one ends up here, and so forth, all the way through the chromosomes. And so here, there's only 6 chromosomes. And interestingly, we can calculate the number of possible alignments based on the haploid, number of chromosomes. So what we do is take 2 and raise it to the haploid number of chromosomes. So here, for instance, we have 3. The haploid number is 3. Right, there's 3 pairs of chromosomes. We split the chromosome pairs in half and we have 3 chromosomes. So 2 to the 3, as you know, is 2 times 2 is 4, times 2 is 8. So you can see on the screen 1, 2, 3, 4, 5, 6, 7, 8 different possibilities.